purpose of this lecture is to provide foundation material for the concepts associated with bioenergetics. The key learning objective in this lecture is to understand the biochemistry that is the basis for energy production during exercise. This concept will be discussed more during the in-class lecture. During this lecture, you will be given the opportunity to complete a series of self-test questions to assess your understanding of key concepts. Your responses to these self-test questions will not be included in your semester grade. This is an outline of the topics that we will discuss in this lecture. We will first start off with some key terms associated with bioenergetics. We'll then talk about different units of energy measure. We'll talk about adenosine triphosphate, also known as ATP. And then we'll discuss the role of the thermodynamic laws in bioenergetic processes. And finally, we'll wrap up the lecture talking about various aspects of chemical reactions, including types of chemical reactions, enzyme coupling, the role of enzyme catalysts, the Q10 effect, and oxidation-reduction reactions. Each of these will be elaborated on in subsequent slides. You can revisit the concepts in this lecture using the navigation bar at the left of your screen. There are several key terms that are central to the study of bioenergetics, but also the larger field of exercise physiology. It is important to remember these concepts throughout the semester. Bioenergetics is the study of energy flow in a biological system. For the purposes of this class, the energy flow we will be tracking is associated with a molecule called ATP, and the biological system is the human body. Homeostasis is defined as the maintenance of a relatively constant internal environment at rest. This term can be applied to almost any physiologic variable. For example, at rest, heart rate remains at a relatively constant level. The typical resting heart rate is between 70 and 80 beats per minute. Also, please note that relatively constant means that the selective variable is fluctuating about a mean value. Steady state refers to the relatively constant internal environment that is achieved during exercise at a fixed intensity. For example, if you start running on the treadmill at 6 miles per hour, after about 3 to 5 minutes your heart rate would reach a steady state. It is likely that your heart rate response would be between 150 and 170 beats per minute and fluctuate about a mean value. All of the physiological properties that we will be discussing this semester involve the use of what are referred to as negative feedback loops. In a negative feedback loop, there is a negative change in a variable that initiates a response. In the case of heart rate, when you start exercising, the body detects that there is not enough blood flow. To compensate, the body increases heart rate, resulting in an increase in blood flow. As we progress through the semester, try to identify other body processes which include a negative feedback loop. A calorie, or cal, as measured in a biological system, is the amount of heat energy needed to raise 1 gram of water 1 degree Celsius from 14.5 to 15.5. In humans, energy is expressed as kilocalories, or kcal, where 1 kcal is equal to 1,000 calories. In humans and many other mammals, adenosine triphosphate, or ATP for short, is the most important high-energy storage molecule in the body. An ATP molecule is composed of 3 parts. The first part is the adenosine backbone, illustrated above by the purple markers. Adenosine is not metabolized, but rather serves as a foundation to bind phosphate groups and store free energy. The second component is the 3 phosphate groups, which are bound to adenosine and each other. The bonds between adjacent phosphate groups are the storage location for free energy. The first bond, which is located between the 3rd and 2nd phosphate groups, is the most important in terms of exercise. Contracting skeletal muscle will split this bond, releasing free energy and allowing for contraction. Once the bond is broken and free energy has been used, you are left with an adenosine diphosphate molecule, or ADP for short, and a free inorganic phosphate molecule, abbreviated as PI. The ADP and PI will then be processed in one of many metabolic pathways to reform ATP using free energy derived from food. There are two more bonds that can be used to release free energy. However, they are typically not used because they store less free energy than the first bond, and the result is the formation of molecules such as adenosine monophosphate, or AMP, and adenosine. These two molecules cannot be readily metabolized back into ATP. Given these two physiological facts, further reduction of the ADP molecule is not favorable, and thus it rarely occurs. The thermodynamic laws govern energy flow in biological systems. I will provide additional examples of how these laws apply to exercising humans on subsequent slides. The first law states that energy is never created or destroyed, it merely changes forms. An example of this is the digestion of food in the gastrointestinal tract and the subsequent metabolism in skeletal muscle to form ATP. The energy in the food has been transferred to ATP, but no new energy has been created or destroyed. The second law states that all physical processes lead to an increase in entropy. Another name for entropy is disorder. In the case of humans, this law is used to describe metabolic efficiency. Metabolic efficiency is a measure which describes how much of the energy in a given food is converted to ATP. Humans, like most warm-blooded mammals, are relatively inefficient. The third law involves properties of crystals at absolute zero. However, since humans are not made of crystal, there is little point to discuss it here. I did want you to at least be aware that there are three laws, but only two really have biological relevance. This slide is meant to demonstrate the first law of thermodynamics. In this slide, you eat a food. The energy in that food is broken down into macronutrients, such as carbohydrate, protein, and fat, in the gastrointestinal tract. After physical digestion, macronutrients are absorbed in the bloodstream and absorbed by skeletal muscle, where metabolism continues. The primary macronutrients metabolized further are fat and carbohydrate. Protein is only used in extreme starvation situations. Once in the skeletal muscle, fat and carbohydrate are further metabolized by bioenergetic pathways, resulting in the formation of ATP and heat. ATP is a storage vessel for free energy, which is what the skeletal muscle uses for mechanical work. And mechanical work in the case of exercise is muscle contraction. The heat that is produced, among other things, contributes to your normal body temperature of 37 degrees Celsius. During exercise, there is an increase in ATP production, but also increased heat, which can become a limiting factor if you're exercising in a hot, humid environment. This slide is meant to outline the principles of the second law of thermodynamics. The second law is really an extension of the first law of thermodynamics that gives additional emphasis on metabolic efficiency and entropy. Since we know that energy cannot be created or destroyed according to the first law, that means that all the energy in the macronutrients must end up as either entropy or ATP. Entropy is a term which means disorder, in contrast to ATP, which is considered to be order. In the human body, only about 25% of the energy found in macronutrients ends up as ATP, with the remaining 75% being lost as heat. In contrast to humans, reptiles are generally very metabolically efficient. They convert the vast majority of energy in macronutrients to ATP. It is obviously not 100% efficient because this would violate the second law of thermodynamics, which governs all biological systems. Now that you understand the laws of thermodynamics, we will consider the properties of chemical reactions, which are governed by those thermodynamic laws. For the purposes of this course, there are two general types of chemical reactions we will consider. Endergonic reactions are ones in which the products have more energy than the reactants. These reactions are considered to be unfavorable because they require energy to be added to the reaction. When we discuss bioenergetic ATP production in class, make sure and pay close attention to reactions where ATP is being used. The use of ATP demonstrates an endergonic reaction has occurred. Exergonic reactions, in contrast, are ones in which products have less energy than the reactants. These reactions are considered to be favorable because they release free energy and do not require any outside source of energy. When we discuss bioenergetic ATP production in class, make sure and pay close attention to reactions where ATP is being produced. The production of ATP demonstrates that an exergonic reaction has occurred. The term coupled reaction denotes the linking of endergonic and exergonic reactions to facilitate the goal of a particular metabolic pathway. This concept is very important because it is only via coupled enzymatic reactions that the unfavorable endergonic reactions are possible. In order to control the rate of ATP reformation, the body has a series of enzyme catalysts. Each enzyme catalyst is responsible for a unique chemical reaction in a metabolic pathway. Some of these reactions are reversible, meaning that you can either form products or reactants. However, a handful of these reactions are not reversible. The reactions that are not reversible tend to involve the use or formation of ATP. During our in-class lecture, make sure and pay close attention to these reactions because they play an important role in the overall metabolic process. The key to enzymes is that they can speed up or slow down naturally occurring reactions. They do not have the ability to cause a reaction that is not naturally occurring to occur. The red line above demonstrates the energy flow in a metabolic pathway from reactants on the left to products on the right. Based on what we learned in the previous slide about reactions, what type of reaction do you think this is? This is a reaction where the reactants have more energy than the products. If you guessed exergonic, you're right. This is a chemical reaction where the reactants have more energy than the products. In this example, you see that the potential energy level must actually increase to a fairly high level before this reaction can occur. The difference in energy between the reactants and the peak potential energy is what is referred to as the activation energy. The greater the activation energy, the more difficult it is for a reaction to occur. If it is more difficult for a reaction to occur, this means that the rate of the reaction will be very slow. This is where enzymes become important. If we add an enzyme into this mix, then this will reduce the activation energy. Reduction of the activation energy makes it easier for the reaction to occur. An easier reaction occurs at a faster rate. A practical example of the role of enzymes during exercise is that when you initially start exercising, your body activates enzymes associated with several ATP-producing pathways. The activation energy of these pathways is reduced, and the rate of production of ATP is able to increase in order to meet the demands of exercise. The Q10 effect is a physiological principle that relates to how enzymatic reactions work in the human body. The principle states that for every 10 degrees Celsius increase in temperature, there is a relative doubling of the rate of enzymatic reactions. When you exercise, you increase your ATP production and result in heat production from entropy. The heat increases muscle and body temperature, which contributes to further increases in ATP production above that's already realized at the start of exercise. While the orange line represents the Q10 response, and it spans a large temperature range, in humans the temperature range is confined to an area between 35 and 40 degrees Celsius. In general, when body temperature exceeds 40 degrees Celsius for an extended period of time, heat exhaustion and heat stroke can result in death. We will discuss heat-related illness later in the semester. Oxidation and reduction reactions play a key role in the metabolic production of ATP during exercise. For now, we will discuss the biochemical principle involved, and then revisit these concepts during the in-class lecture. During the in-class lecture, pay special attention to the transfer of electrons during the TCA cycle and the electron transport chain. The understanding of oxidation and reduction is key to explaining how the body reforms ATP using aerobic means. An oxidation reaction is one in which a molecule loses a pair of electrons. Loss of electrons means oxidation. Reduction, on the other hand, is a process in which a molecule gains a pair of electrons. So gain electrons, reductions. Lose electrons, oxidation. This concludes your introduction to bioenergetics. The information presented in this lecture was meant to prepare you for advanced information that will be presented during the in-class bioenergetics lecture. Make sure and complete the bioenergetics quiz prior to the in-class lecture. Please post any content-related questions that you have in the discussion forum on Blackboard.